Okay, so record is recording now. Okay, so welcome to the 2025-2026 seminar series of the AAAC, so Association for the Advancement of Affective Computing. So it's the third we have. And it is, well, this is part of the Education and Early Career Committee. And it's my pleasure to open this year's series with Professor Jeffrey Krichmar, Jeff for the people who know him. And let me just read a little bit what the bio that we prepare here. So he received BEc, BS, MS and PhD in computer science, computational sciences. And after that, after his PhD, he spent 15 years as a software engineer on different projects, like at IBM and other places. But in 1999, then he returned to academia to do something different. So he was a senior fellow in theoretical neurobiology at the Neuroscience Institute. And I guess this will be very obvious in his work. He also has a very broad knowledge of neuroscience. Since 2008, he has been a professor at the Department of Cognitive Science and the Department of Computer Science at the University of California, Irvine, where he's been designing adaptive algorithms and neurobiological plausible neural networks and brain-based robots, basically. And he is very well known for his work on neuromodulation and his publications. He has over 150 and 9 patents, and that's impressive. Okay, so today he's going to talk us about neurorobots and neuromodulation and the interplay between affective learning and behavior in the brain-body environment loop. So the floor is yours, Jeff.
Great. Thank you, Lola, for the nice introduction and inviting me here. And thank you all for attending. Yeah, I'm going to talk about neurorobotics, which is a field that's our lab is really focusing on. And as Lola said, I've done a lot of work on neuromodulation. So I'm going to talk about how that relates to neuromodulation. And most of the examples will be from our own lab. There are other labs that are doing this, and I'll touch on a few of those. But first, what are neurorobotics and what's the approach? So as Lola said, I'm a computational neuroscientist, so I like to make brain models, but the brain needs a body. So the idea of neurorobotics or cognitive robotics is the robot controller is modeled after some aspect of the nervous system. And the power of this is twofold. Once you have this set up, you have a closed loop system and you can test theories of neuroscience in a more embodied way, in somewhat natural way. Hopefully some of the examples I show will tell you that. And then also, even though there's been lots of advances in artificial intelligence in recent years, they still aren't the same, and I don't think are as impressive as biological intelligence. They don't have the same capabilities of biological intelligence. So this might be a way to actually build autonomous systems with some level of biological intelligence. Now, I've been doing this for a while, teaching it for a while at the university, at UC Irvine, University of California, Irvine. And my former student and I taught this course called Cognitive Robotics. And eventually we realized what we wanted to teach was not in any textbook. So we wrote our own book. And so a lot of what I'll talk about today follows this book called Neurorobotics, Connecting the Brain-Body Environment. And at the heart of the book, there are these design principles that we lay out. And hopefully you can see my mouse. We're going to go there. There are three levels. I think for this group, I'm going to concentrate mostly on the learning and memory and the behavioral trade-offs because that's where neuromodulation and affect really show up. But let's briefly talk, because it's absolutely essential, about the first level of design principles, which is this embodiment and reactions and kind of responding to what's happening right away. And there are many elements. I'm just going to touch on a few just so you get a taste. So there's this concept by Rolf Pfeiffer and Josh Mongard called morphological computation. And certain processes performed by the body free up the brain to do other things. And the brain's pretty slow. So the body and actually the maybe short loose between the spinal cord and vertebrates uh, actually respond quickly to events that happen in the world, faster than the brain could probably keep up. The brain's very good, or the central nervous system and the cortex is very good at, predicting what's going to happen, planning ahead, and then if things don't go as planned, then adapting. But the body, to free that up, the body does a lot of things. And uh, this nice anime shows, or is it manga? It shows that, with all the walking robots are really take a lot of energy. They have a hard gait and they take a lot of computation. Whereas dogs and this robot that looks like it's an insect just kind of scramble over things and they don't even have to think about it. And we do the same thing. And we also use things like gravity and friction to our advantage, whereas the robot on the far right actually is using it, trying to get around that with computation. So here's a couple examples. On the left, we had a project where we were supposed to play soccer, or I guess you're in Europe, so it'd be football. And we had a really good vision system. It couldn't catch a ball. It just came too fast. And so eventually we realized that, well, what if we use the body? So we actually put this plastic loop around the robot, this little thing that traps the ball. And then the robot does have sensors to feel what's hit it and then turn around and look at it. And that, thing that costs very little money. So it's a cheap design, cheap in efficiency, um, solved a difficult problem and freed up our vision system that we had a large neural net for a vision system from having to do all this heavy lifting, but also it just wasn't fast enough. So that's one example of morphological computation. Another example is in grasping, which has become quite a important topic now because a lot of people are designing humanoid robots to do household tasks, but they still have issues with grasping things. Um, and the pose of a hand is difficult. But if you have soft compliant materials like in nature that can just wrap around whatever is in front of it and then have some feedback or sensors that just say that, oh, I've touched something, it could pick up a wide range of shapes, sizes, weights, um, and also depending on how fragile the object is, because it doesn't have to, it just softly goes around it, so it doesn't have to push too hard to actually pick it up. So that's another example of embodiment morphological computation. And this also doesn't just occur at the periphery, it occurs in the brain. And there's a whole field that I am a part of called neuromorphic computing. And the idea of the field was to look at how the brain actually computes and develop hardware that takes advantage of that. And so neurons in the brain, they spike, right? And why do they spike? Well, there's arguments in neuroscience. It's a rate code, it's a temporal code. Uh, but I think the reason, and a lot of people think the reason neurons spike throughout nature is because it saves energy. So what happens is during sub threshold kind of integration at a neuron, there's not much energy expended. And then when it goes above threshold, it outputs a spike. So it turns an analog signal into a binary signal. And then that takes some energy, but the real energy consumption is when that signal is received by a neuron through a synapse. And that, is neurons fire at very low rates. So what you have then is a lot of energy savings, and you have massively parallel because you got all these neurons like their own little computer. Um, and it's event driven. So you only respond when an event comes. So there's no need for a clock. And this is not lost on a lot of hardware engineers from Intel, IBM, um, groups in universities like Manchester building the Spinnaker. So let me get through the sound for this. So this is Tiffany Hu and a group from IBM with their True North neuromorphic chip. And this was the first time they tested in a closed loop. It had tested, so it had trained with a neural net to recognize a trail versus non-trail. And then we strapped this chip onto our robot with Velcro to see if it actually works on these mountain trails in Colorado. And it's a very steep trail, so that's why we have the rope, because this was a one-off computer. They made more eventually, but we didn't want it tumbling down the mountain. And it's a simple neural net in the grand scheme of things, but what was impressive to me was we ran up and down, and you can't really tell with this video how steep and rugged this trail is, but we ran up and down this trail for an hour on the small nickel metal hydride robot powering a small nickel metal hydride battery powering the robot, and we also set up the power to the chip, and it continued to run. We never ran out of power. So really, the take-home lesson for me was these things could actually be really great for edge computing. And the reason is because they, like the brain, are event-driven, massively parallel, and save a lot of energy. So I think that's another way of looking at the embodiment and also what we call efficiency through cheap design. Oh, I should say, I'm not going to be looking at the chat. If you have a question or need clarification, speak up, or maybe someone can just holler that, oh, there's a question in the chat. Otherwise, we can have discussion after I'm done. Okay. So I only briefly, I mean, I could go, in fact, last night in my class, I went for over an hour just talking about this first level of principles. But let's talk briefly about the other two, because I think they're more pertinent here. So how about over a lifetime, we have to adapt. We have to learn things. We have to remember things. And what we need to do that is some sort of set of values. And it's not just good and bad, all right? It's sometimes what's interesting and or curiosity or how much I want to attend to things. And when I started looking more deeply into value systems, kind of when I was transitioning from the Neuroscience Institute to UC Irvine, I started reading about these and kept coming across these neuromodulators. So neuromodulators in the brain, you've probably heard of dopamine and serotonin. And then there's a few others. The four major ones people usually talk about are serotonin, dopamine, norepinephrine, and acetylcholine. And what I found was interesting is they're all subcortical. So they're all, in the midbrain or brainstem, and the sources of all these neurochemicals are in these nuclei. And then if you follow the colors, they have broad signals across the brain. Um, the other thing, it's not just going into the central nervous system. They also, or not just the cortex, they also have broad signals down to the spinal cord and the periphery. And I should say these are a lot of times bidirectional. Now, when I looked at it more, and this is, 2008, I said, well, they all have certain events they're looking for. Like serotonin, you could think of the cost of an action. Dopamine's well studied about reward prediction, predicting your expected value. Um, Norepinephrine really triggers when something surprises you. And so you have something unexpected. And acetylcholine is very important for learning, but also how much effort you want to put in with your attentional system. But what all of these have is they're tonically active so that they're always putting out some level of this chemical into the brain when you're on task. And then the behavior seems to be kind of curious and maybe a little random. But then if one of these events happens, then there's this very quick phasic response and the organism is decisive. Um, so we call it exploitive. So it's an exploitation trade-off with these things. And like I said, they're the main one we think about is reward and punishment. And this is a nice review by Bro and Diane that relooked at the opponency between dopamine and serotonin. And it's kind of a subtle opponency, but dopamine does signal reward and serotonin does signal some sort of punishment or cost. So this is going the way back machine. This was my first Neurorobot. We were not allowed to call it neurorobots. We called it brain-based devices when I worked with Gerald Littleman at the Neuroscience Institute. He had a theory called neurodarwinism. So my first robot was the seventh iteration. So it's Darwin 7. And it had back in the day, it had a very large scale neural network with auditory streams, visual streams, um, and a value system. Now, the robot's task was to look for these blocks and taste the blocks with its fingers. And the taste of the blocks was how conductive they were. So it was highly conductive metal. It was good tasting. It was weakly conductive metal, like over here, it was bad tasting. And the stripe pattern would be good tasting. And the spotted or blob pattern would be bad tasting. And it learned which which patterns were predictive of good or bad value. And just to show you my first neurorobotic experiments. This is Darwin 7. This is its early visual system. So it's got retinopathy, and each one of these pixels is a neuron in its brain. And this is a model of area IT that is responding to the whole object. And then down here is its value assessments. On the left would be bad tasting, on the right would be good tasting. So this is the first time it's seen the stripe pattern, picks it up, it's good tasting, it's building up a representation for this. This is the first time it's seeing this spotted pattern. So it's going to pick it up even though it's bad tasting. Note the activity on the left side here. And after about 8 to 10 pairings, before I run the video, it's approaching this object, even though it's slightly turned. It's got invariant object recognition. There's a strong, clear pattern in that area IT that's enough to drive the prediction that this will be good tasting. And so then picks up the good tasting block, very strong pattern. Approaches the bad tasting block, but then won't pick it up. And so this is an early example of reward versus punishment opponency using neuromodulation. And once you have these values and these modulatory signals, they really boost learning. And the way we learn is much different than AI. With just a few presentations, we can learn something new, an object or a new skill. And we're very good at when we learn new things, we're okay at it. We don't forget what we learned before, even though that's a big issue in artificial systems with catastrophic forgetting. And then we can take what we learn from one situation and apply it to another, which is another hard problem in artificial systems called transfer learning or generalization. So how do our brains do that? And I could spend, and I don't know all the answers, and I could spend hours talking about memory systems. But one thing that Tiffany and another student in our lab, Harak, and I looked at was the idea of schemas and context. So this is a restaurant in Irvine near our university. And I've been there and I'm familiar with the restaurant, though it's been a few years since I've gone to this particular restaurant. Um, and this is a restaurant in Seattle, which I haven't been to. But if I went to it, I wouldn't have to learn everything all over again. I have a schema or context for what happens in a restaurant. I know when I go to the restaurant, someone's going to seat me at a table. So there's going to be a table and chairs. They're going to hand me a menu. Eventually, a waiter or waitress will come over and take my order, and hopefully I get food and it tastes good. So I have this schema of what a restaurant is. That allows me to learn something very, very quickly because the old idea in episodic and declarative memory was the hippocampus is involved when you learn something and gradually over time, and it takes a while, depending on the organism, it gets consolidated in the neocortex. But they found out that animals and humans, when there's something with a schema, you can get that information consolidated very quickly. So we made a model of the hippocampus, that's this HPC, and an area that was known to be involved in schemas, the medial prefrontal cortex. And we would give a context to this model, uh, some sort of pattern, and I'll explain that when I run the video. And the uh, the system, the neural network, would learn that this is a particular schema. So if something was um, unfamiliar and novel, you'd have to create a new schema. And so we had these neuromodulators. Tiffany, when she designed this, just called them neuromodulators, didn't, but there's evidence that there's probably dopamine and norepinephrine are involved with these kind of signals. Now, if it was novel but familiar, it might fit within a schema. So you would actually, instead of creating a new schema, add this to your existing schema. And then the action was, you'll see in the video, to actually go where this object might be. So let me start the video now. So if we type in an object like teddy bear, and then our robot is roaming around a room that's a classroom, and not only is it looking for a teddy bear, but it's building up a schema of what's in this room. So it builds up a classroom schema. Finally finds the teddy bear. A few iterations later, it knows trial 4, it knows right where the teddy bear is and picks it up. Now we put in a new context and say, retrieve a cup. And so it realizes this is totally unfamiliar and novel. So it creates a new schema and it's looking for the cup, but it's also building up a schema uh, on this environment. And the key is well, here's the 4 trials later, so it knows exactly where objects are. But the key is, what if we put it back in the classroom and ask it to pick up something it's never picked up before? It does that immediately because it loads in the original schema from before. And then if we have an object that it's never picked up before, like a banana, it says, well, which Which schema would be that? Well, it would be in the break room. There's always food on the table. So those are the likely places that you should find a banana. And this provides a clue like that maybe having this notion of schemas in the brain and controlled by the frontal cortex actually might be a way the brain actually gets around the problem of catastrophic forgetting. And because of this group, we're talking about neuromodulation and affect. We struggle with this network for a long time till Tiffany said, why don't we put it in? We should have done this at the beginning, given what we've done in the past. But why don't we actually have these two kind of knobs of neuromodulation, depending on these two concepts of familiarity and novelty? And then once she had that in as inputs into the model, then things started to work really smoothly. Okay, the last set of design principles is called behavioral trade-offs. And this was kind of unexpected, but over the years when we read about these things, we realized that a lot of things that happen in the world, it's a trade-off. And there's, depending on the context, it can push you on one side or the other. We've talked about reward and punishment. We've talked a little bit about exploration, exploitation, but other things like are you invigorated or withdrawn? Are you stressed? Are you calm? Do you want to forage, which you need food, but then you're leaving your nest available, so do you defend? What's interesting is a lot of these trade-offs are regulated by neuromodulation and also hormones. I'm going to go through a few of those. But we talked about reward and punishment and dopamine and serotonin. It's also involved in risky versus conservative behavior. I'll talk a little bit about attention and some of these, and then but some of the other things I probably won't talk about today. Stress versus calm. There's some hormones, glucocorticoids, in your brain that actually can trigger, depending on the level of these things, pushes you one side of this trade-off or another. So let's go back to this Bourreau and Diane diagram, and now let's look at the vertical axis, invigoration versus inhibition. So dopamine, when there's a lot of dopamine in the system, you actually are more invigorated and you're novelty seeking. Serotonin, it's complicated. When serotonin's involved in the system, I won't say high or low because serotonin is complicated with the way the receptors work, but it can lead to inhibition or withdrawal behavior. And you can imagine this happens during anxiety or depression. So I thought this axis was interesting to explore. And one way people, and this is typical of our field, if you want to look at something, you look at, well, with a robot, you look at, well, what was the behavior that was used, as the gold standard, for example, in the literature, either the human literature or the animal literature. And for anxiety tests and curiosity, there's one of the paradigms that people use is called the open field test. And typically with a mouse or a rat, you put it in an open field. It's usually darker along the edges, so it'll start along the edges of an environment. And a lot of times they give them a little home nest and it'll kind of retreat to the nest. But then eventually, when it realizes that this is a safe area, it will actually make excursions and cross over the middle. And then if there was an object in the middle that was worth exploring, it would actually explore it. And people have used this test in serotonin, shown that when you mess up certain serotonin inhibitors genetically, that there's less time spent in the center, that there is less time approaching a novel object. And yeah, or actually not less time, takes more time before you approach the novel object. The distance traveled, because you're kind of withdrawn, goes down too. So it really shows this inhibited and withdrawn behavior that can change if the serotonin system is impacted. And the dopamine system, um, things like cocaine, which they put on animals, increase dopamine levels in the nervous system. And in the open field test, that means that there's more activity. The rat is invigorated, and it actually runs more in the center. It's taking more risks. So given these kind of background, there's a whole bunch of background on this. We designed we designed an open field test for a robot. And so we call all our robots Carl. I should have said that at the beginning. We're the Cognitive Anteater Robotics Lab, so it makes for the acronym Carl. And this was Carl built on a Roomba, so it's called Carl Roomba. And the robot is now doing this wall following. I got to turn that off. And it's got the Roomba charging station in the upper right corner of this environment. And so eventually the robot system says, all right, this is safe. No predators around right now. So then it crosses over to the middle and explores this novel object. All right, so nice example of a robotic open field test. And we made a model based on some of these ideas of neuromodulation. So we had an attentional system. So if something occurs, like there's a novel object, a flash of light, or the robot bumps into something, if it's one of the first times, it'll attend to it. If it happens over and over again, it attenuates or habituates to it. And then there's four things it can do. It can do exploring an object or crossing into the middle, and those I call our curious behaviors. Or it can do wall following and finding its charging station. I call those anxious behaviors. And dopamine is something that if there's dopamine levels are high, it'll tend to do those curious behaviors. If the serotonin levels are high, it'll tend to do the anxious behaviors. And a bump, if it's curious, can either be something to explore, or a bump, if it's anxious, be something to run away from. So here's a neural trace of the experiment. And we run this for several minutes, and you'll see that initially serotonin levels are high. There's a lot of anxious behavior. But over time, the serotonin levels kind of come down and dopamine levels start to rise. And so then it starts to tend to explore. And around the halfway point, there's a flash of light. And so the flash of light is something unexpected and also potentially dangerous. And you see this bright red band here. So that's a phasic response of the serotonin system, which causes it to immediately run to its charging station. And so what we can do with these is change the level of both how active these neurons are, or when a neuron releases serotonin or dopamine, uh, eventually it has to be taken back in. So it's called reuptake. So we have time constants for how fast it reuptakes, so we can play with that. And why that's interesting is here's the control experiment. So where we ran this a bunch of times, and after about 60 seconds after the flash of light, so zero is the flash of light, you see very anxious, scared behavior. Sorry for anthropomorphizing. And then around 60 or 70 seconds, dopamine starts to come back and trigger curious behavior to actually explore. So if we change the reuptake, so there's a lot of serotonin in the system, after that flash of light, the poor robot is never out of that anxious state. So it's got almost like an anxiety attack, or it's hiding by its charging station, so it's in sort of a withdrawn state. And if we increase the dopamine with that playing with the amount of reuptake, then we have a robot on cocaine, if you will. And that robot is fearless. And after the light, it doesn't matter. It's constantly exploring and going to the middle of the room and looking at novel objects. And so in a sense, it becomes obsessive compulsive. And not to be cute, but I mean, this is actually a nice test bed for testing neurological disorders. And also we can match it, both the neural activity and the behavior to experiments with animals and make some interesting predictions. Okay. And serotonin, I said, was complicated and interesting. I went to visit Kenji Doya's lab a number of years ago and talked to the Miyazakis, and they said, serotonin also is important for patience, uh, not impulsivity, but patience. They were very clear about that, and they said, they had a hypothesis that um, if you have rewards that vary, so there's a lot of uncertainty of the rewards and when they're coming and how big they are, then you actually, serotonin becomes into play, especially how long you'll wait. So let's look at B and C. You have a reward that comes at 4, 6, or 8 seconds, or 2, 6, or 10 seconds. Um, how long will you wait for that reward or just give up because you think it's not going to come? And they actually did this work with mice and were able with optogenetics to increase, to activate the serotonin system. So when serotonin was activated, these blue lines, it actually would wait longer for that reward. Whereas when the serotonin was not activated, it would shift to the left and not wait as long. So having more serotonin in the system made the organism more patient and waiting and less, I hate to say, I guess the opposite would be impatient uh, for for uh, for a reward, and then it would just give up and do something else. So I thought this was really clever and beautifully designed. And actually, when you got down to it, it was a simple concept to code up and put in a neural net. So we did this with our one of our robots, and this is low serotonin, so the probability of waiting drops off around this time period in seconds. And then if you had high serotonin, you can push this curve out and make the robot more patient. And first, let's show the low thing. So the task of the robot was to head to all these waypoints. And if it has trouble finding a waypoint, it gets impatient, just like the mouse saying, "Oh, I'm giving up on trying to wait for this reward," and it jumps ahead to the next waypoint. But now, if we take the same robot and same task, but then increase the serotonin, and now you look at the one on the bottom, it's going to work hard to get to these waypoints. And in that sense, it's patient. This was not part of the design, but there was a child, because we did this in the park, and this child was very excited about the robot. And so we were like, "Oh, don't touch the robot, don't touch the robot." But anyway, it was also patient with the child, but that was not coded in. But I thought this was interesting, and this is the raw trajectories under high serotonin, trying to find all the waypoints along this little road here, and then low serotonin, and over by these trees here and here, it was difficult to find waypoints, so it would give up and shift over, which actually is not a bad strategy for robot exploration. And so this is a nice way to actually smoothly um, have a trigger or a knob to play with how assertive you want your robot to be, and could have interesting implications and also interesting implications on how the serotonin system works. Okay, I haven't talked about much about acetylcholine and noradrenaline and the attention system. So this is a subtle trade-off. So in our attention, we have, if we're in a noisy situation, we kind of have an expectation of how much noise that is. And this is sometimes called in the literature expected uncertainty. So the known unreliability in the environment. But then if something happens that we weren't expecting, a surprise, an oddball, uh, you get this unexpected uncertainty. It violates prior expectations. And noradrenaline the noradrenergic system seems to be responding to these unexpected uncertainties. And so you have this interesting nuanced trade-off in the attention system. And this was first, explored by Angela Yu and Peter Diane. And you'd have a cue, and oops, sorry about that. You had a cue, and one of these cues was predictive of the light. Okay. Um, and in this block, it might be the blue one. But the cue had a certain validity, right? How uh, it could be like 90% in other experiments, it could have been 80% of the time it actually predicts, and the others it doesn't. And then without telling the person, uh, then you change the cue. You change the cue identity. So all of a sudden, without knowing this, it would suddenly become red. And so it would have a new cue, but it also would have a certain validity too. So this was this trade-off between expected and unexpected certainty. And they showed that norepinephrine and acetylcholine were tracking these two things. And this actually they did this in a model and some experiments, but we thought, hey, this might be actually interesting in a neural network model. So first, in the upper right, we did this with MNIST digits, those hand-drawn digits. So we would show the system pairs of these. And the system would have to guess, is the Q either it's a high number or a low number, or is the Q is it an odd number or an even number? And so, for example, odd was the goal. So it had to figure out odd is the right Q, but odd was only showing up 85% of the time. And so you see in the acetylcholine uh, a lot of noise because it's tracking the level of expected uncertainty. But as soon as that Q changed from odd to low, you see this spike in the noradrenergic system saying, hey, that was unexpected. So there's a phasic response, and boom, then it kind of throws out the priors and tries to build a new expectation level. And so this is how we think this attention system works. So you say, that's cool. Let's put it on a robot. So this is work by Xinyin Zhou, and the robot is trying to guess what the user wants. And the user is actually, from my perspective, I'm sitting at a desk, and the goals could be either say hello, there's Xinyin saying hello, read a book, do some work on the laptop, or eat something. And so depending what its expected uncertainty is, it's building up what is the most likely thing to pick up. And eventually it decides the person's goal is they want to eat. Sorry for the slow robot. And then it goes and picks up an apple. And it'll bring the apple to the user, which would be the person videotaping. Here's what I said about grasping before. And then the person will give feedback to the robot. Yeah, I was hungry. Thank you. Or so that would be, and sometimes it wouldn't. So that would be your expected uncertainty. And sometimes it'd say, no, I didn't want that. And the robot has to guess. And so then you see the same pattern with the robot's neuromodulatory system, where it builds up these expectations based on the expected uncertainty in the cholinergic system. And then it has these responses when the person changes their mind. And you can see how very fast it switches actions of what the user expects. So not only is it a nice example of an attentional system, based on neuromodulation, it actually could have practical applications for human robot interaction where you don't always know what the person wants and needs, and you have to respond quickly when they change their mind, which people will do. Okay. I've said before that, or I might have said, should have said, I guess I did say before, that all of these neuromodulators are triggering this exploration versus exploitation trade-off. And when we started looking at this in 2008, we said, yeah, that um, there's this difference between the phasic and tonic activity. And so that's worth exploring on its own. So this was the first Carl robot, and it was really looking at the phasic neuromodulation. And before I run this, you see the pixels in here. Each pixel is a neuron in its brain. The level right now is tonically active. It responds to colors, green, cyan, magenta, red. And it's got a dopamine system. VTA is the ventral tegmental area for dopamine. RAFA is the serotonin system. And like before, if the neurons are bright red, then they're highly active. If they're dark blue, then they're off. And we had this disco floor that the robot explored, and it could give signals of reward or punishment. And it's now learned that red is going to be punishing. And you see this strong signal from the serotonin system. But when it flashes green, there's a phasic response of dopamine saying, hey, that's predictive of reward, and it goes and seeks it. And in this way, we were actually showed the phasic tonic relationship. One thing we didn't expect was when you have this um, phasic neuromodulation, that you actually have an incredible signal to noise ratio boost in the visual system, because it actually gives a signal to actually turn on what is important, be decisive, and turn off everything else. And so that actually has a very rapid response, but also a very high attentional effect. And then when it's something that's non-salient like magenta, then it goes back to tonically active. Okay, I think this is my last trade-off. How am I doing for time? Oops. Oh, all right. We'll wrap up soon. I did want to touch on this because it's just to say it's not just modulators, it's also hormones are having these trade-offs and having these effects. One of those is oxytocin. And oxytocin is very important for pair bonding. It increases in parents to pair bond and take care of their child. It increases in the child to actually bond with the parents. And this is a rodent brain, and this is also in the rodent brain. And here's some of the pathways for oxytocin. So there were some really interesting experiments by this Lab Young and uh, all his students that have gone on to further study and start their own labs. And they worked with these voles. And I can't really tell the difference too well, but the prairie voles are monogamous. Once they mate, they stay together, and both the mother and father take care of the babies. The meadow vole is polygamous. So after the male mates, it takes off and leaves the female to take care of the babies. And they looked at monogamous moles versus polygamous voles, and saw there were more oxytocin receptors in areas like the reward areas, prefrontal cortex and stuff. And then also there's another hormone that's involved in this, the vasopressin, which actually there was difference between the monogamous voles and polygamous voles. And then they actually could do some genetics and make a monogamous vole into a polygamous vole and vice versa by doing a receptor antagonist to oxytocin to actually have it not bond as much or antagonist to the vasopressin receptors for that hormone. So interesting that how the level of this hormone or the level and actually across species changes and actually dictates or has an influence on important behaviors. And this is not lost on the work from Lola Canamaro's group over the years. She's done pair bonding with robots and among other things, and actually looking at some of these hormone effects. And I won't try and explain this too much because Lola would probably do a much better job than me. But you should definitely look at her work over the years on these systems and how it affects behavior and how it the feedback and interplay between these systems works. Okay, so let's wrap up. And for the behavioral trade-offs, the world is full of contradictions. Uh, and it affects all these different trade-offs. And it's interesting that it seems like hormones and neuromodulators in the vertebrate brain at least trigger or regulate the balance of these things and which side of the trade-off you might fall on. It's not to say that they rule our lives. There's strong connections from areas like prefrontal cortex and amygdala and other areas to these systems. So they can be, they can have set points and regulate them and actually kind of push it. But it is interesting that these chemicals have such an effect. And I think having these trade-offs in your system actually leads to interesting behavior. Conflict makes for a good story. So actually putting them in the robots, and we've always done this over the last several years, it makes them more interesting and relatable, and more interesting to study, and I think more biologically plausible. All right, I should wrap up now. So I went very rapidly through all of these principles. I think uh, if you're into robotics, I think it's important to consider these when you're building your system. But if you're not, it just gives you maybe a better understanding of what makes something intelligent, or how it might improve an autonomous system. Doesn't necessarily, I mean, I'm very big on the idea of robotics and embodiment, but maybe there's some other system that could take advantage of these ideas. And I'm just the spokesperson. Most of these people here did a lot of the work. I didn't get to talk about all their work, but let's see. There's Tiffany, talked a lot about her work and her rock. And you saw with the patient's work was Jin Wei over here. Um, Been quite fortunate to have a great group of people doing all this kind of crazy kind of studies over the years. So I will stop there and take questions. Thank you for your attention.
Thank you very much, Jeff, for this like really interesting talk with lots of information. And I'm going to upload. But some people are uploading
Oh,
online,
let me
but
look at you all. I can bring back. Thank you.
on behalf of everyone, and I am going to stop the recording so that people can ask questions just without worrying about